We're team number 5, and this is our problem solution to the following disease, Gaucher's disease. In this presentation, we will dive into Gaucher's disease. We will study its causes, metabolic pathway, and treatments, as well as researching about a treatment innovation with the use of biotherapeutics. First of all, inherited metabolic diseases are metabolism malfunctions caused by genetic conditions. Gaucher's disease, also known as GD, is caused by a deficiency of an enzyme called glucocerebrosidase. There are different types of the disease, and they could cause from mild to severe effects on their patients. The pathway of hydrolysis of lactosylceramide is important for the study of GD because it involves the degradation of the enzyme talked about before. Here are some examples of the current treatment for the disease.
Enzymes are molecules, mainly proteins, that bind to a substrate and decrease the activation energy for a chemical reaction, making it faster and more sustainable. Glucosylceramidase, the protein of interest, is a hydrolase enzyme that catalyzes the breakdown of glucosylceramide into glucose and a ceramide.
Next, we're talking about enzyme characterization. Enzyme characterization helps understanding mechanical properties of enzymes, giving clues on how to approach certain pathways, and also help us to predict how an enzyme will work. First one, for the kinetic characterization, the Michaelis-Menten equation can be used using its parameters Pmax and Km that are obtained experimentally, while substrate is controlled. On the other hand, Arrhenius type plot is used to analyze how temperature affects chemical reactions, and it calculates activation energy. When we talk about biotherapeutics, we are talking about antibody, enzyme, or other drug cell therapy products made with living systems. They are considered a great innovation in the pharmaceutical industry because of their successful records on chronic or life-threatening diseases. Purification. Process chromatography is the best way to approach purification. It helps releasing impurities by going through different phases. Not purifying correctly may be a risk for patient safety. And its characterization. Structure, post-translational modification, and cellular mechanisms are features that must be characterized to control biotherapeutical behavior. Types of existing characterization are based on its structural and physiochemical characterization, in its immunological properties, on its biological activities, and in purity and contaminants.
Imiglucerase is obtained by recombinant DNA technology using human acid beta-glucocerebrosidase with mammalian Chinese hamster ovary cells. Two methods of purifying are hydrophobic chromatography on butyl cephalose, where it is used a pH of 6.2, and in the method they use a pH between 5 and 7, so it's perfect. And the cation exchange, where protein expressed by recombinant DNA are typically associated with contaminants, and this is a form of purification that contaminants. Some differences of a chemical treatment and a biotherapeutic is the synthesis. In the first one, it is rely on controlling physical chemical parameters, and in the second one, you have to define what is wanted to be produced and identify the specific gene, cloning to make it produce the required protein, and synthesis occurs inside living cells. The reagents used in a chemical treatments are a lot of solvents and pollutants or toxic for the environment and human health substances. And in the other one, there are like 4-methyl lumbaliferyl, glucoside restriction, and the nucleases, etc. The residues on a chemical treatment are chemical sludges, wastewater, etc. And in the other one, it is recommended to not reuse containers or empty jars of biotherapeutic treatments, and it produces biological hazardous waste that has to be disposed, like I said before. The process complexity in a chemical one is the low specificity, and it might affect other cells. And in a biotherapeutic, they're bigger and more complex than chemical, and their characteristics and properties depend most of the times of their own fabrication process. And the operation conditions, they in a chemical treatment they search for a balance with preservation of normal cells, and the other one is often requires specific but easily achievable parameters of temperature, humidity, and pH. As we know, biotherapeutics, such as the proposed solution for the treatment of Gaucher's disease, are a powerful way to treat diseases that would otherwise require invasive intervention. The use of a recombinant protein ensures that the direct root of the problem is addressed, and there are a lot of methods available for the production, extraction, purification, and delivery of protein therapies, where the most effective and safe methods can be utilized.